So now we're moving on to genetics. And the first topic to talk about in genetics is Gregor Mendel. He is known as the father of genetics. He's the first person to stumble on the basic principles that describe how we inherit our traits from our parents. So genetics was technically born in 1866, but it wasn't formally discovered until about 1900. And I'll get more to that later. Before 1900, most scientists believed in the theory of blending inheritance. And what that theory was is that we were basically haploid, and when we sexually reproduced, we passed information to our offspring. And when a mother and a father passed the information down, it somehow chemically blended and became one set of instructions in the offspring. So we were basically haploid. They called that blending inheritance. Gregor Mendel, born in 1822 in the Czech Republic, entered a monastery and taught high school. He was a high school teacher. He ran a monastery garden. He originally wanted to study mice, but he was turned down for that. So he ended up studying peas in the monastery garden, and he published a landmark pea genetics study in 1866. He succeeded in figuring out the secrets of heredity because he knew the pea plant very well. He chose them wisely. The pea plant is a member of the pea family, and the flower is a flower that he understood. Peas normally in the wild don't self-pollinate, but the garden varieties do self-pollinate due to excessive inbreeding, and that allowed him to control the pollination patterns that were in his garden. He also realized that pea plants have a number of very interesting varieties, and that gave him the opportunity to watch how the different traits for flower color and pea color and pod color got passed from parents to offspring. So this is Gregor Mendel earlier in his life, and this is the study plant that he is famous for. Okay. So what Mendel did was he crossed pea plants of different strains and he produced hybrids. He called the pure strains of plants that he started with his parental strains, and he called them the pea generation. The hybrids were referred to him as the F1 plants, meaning first filial or first offspring. What he first noticed in his study was that instead of blending inheritance, all hybrids resembled either one parent or the other, but not both, and they were not in between. So if he crossed a red flowered pea plant with a white flowered pea plant, he didn't get pink flowered offspring, he got red flowered offspring and no whites. His conclusion from the initial crosses was that inheritance factors do not blend with each other, and apparently some inheritance factors are dominant over others, meaning they can supersede other traits. So he didn't know why red flower dominated white flowers or why smooth pods dominated wrinkled pods, but that's the way it was. Tall pea plants, the vining pea plants, that trait dominated bush peas, which were short plants. He didn't know why that was, but he is credited with discovering the phenomenon of dominance and recessiveness. Now, the next thing Mendel did was he took his F1 plants, the hybrids, and he crossed them with one another just to see what would happen. And so what he found was that in the F2 offspring, that's the offspring from the F1 plants crossing with each other, most of the plants were like their F1 parents, but a fourth were not. So in this schematic here, what you see is a white flowered plant and a red flowered plant. These are kind of representing flower color traits. And when you cross those two, all offspring are red flowered because apparently red dominates white. When you cross these plants, they're not the same as this one. When you cross these plants, most of the offspring, 3/4 are red, but 1/4 were white. Now, Mendel sat down to try to explain why that happened. So Mendel made some assumptions. First, he assumed that the hybrids had the recessive factor. In other words, they weren't haploid. There was no blending. Instead, he assumed that these two plants, when they reproduced, they produced these F1 plants, and the F1 plants had red and white information in them, but that the red was dominant, and that's why they were red flowered. He assumed that the factors for the red and the white would separate when the pea plants made their sperms and eggs. So pea plants only got one of the factors. They'd either get the red factor or the white factor, depending on what the available genes were or the factors were from the parent. So for pea plants, gametes are sperms and eggs, which basically are pollen or ovules. Now, Mendel represented the factors, which we now call genes, with letters. Uppercase for dominant genes, lowercase for recessive genes. And a genotype, then, was the combination of genes that an organism had. The physical features that the organism had as a result of its genotype was called its phenotype. So, if an organism was homozygous, that would mean that it had either two red factors or two white factors. And so a homozygous red factor plant would be big R, and a homozygous white factor plant would be little r. Heterozygous organisms would have two different letters, like big R, little r. They have the red and the white. So this is a homozygous white flowered plant. This is a homozygous red flowered plant. All four of these plants are heterozygous red flowered. These four plants are big R, little r. This one's big R. This one's little r. When you cross a couple of these plants with each other, you get 3 red and 1 white. Now this plant right here, we're for sure it's little r. But as it works out, 2 of these plants are big R, little r, and one of them is big R. But that's for when we do the Punnett squares. Then Mendel took a look at the study of 2 traits simultaneously, just to see if some traits stuck with other traits in reproductive patterns. So he originally studied plants he started out crosses where plants were red flowered and tall, meaning they were dominant for flowers, dominant for height. Okay, so you can use any letters to represent them, but here I've got A's and B's. Then he picked plants to cross with them that were white flowered and short, recessive for both traits. When you do that, the F1 plants are all going to be heterozygous, meaning they're going to be like big A, big B, little b, because they're going to inherit a big A from one plant, a little a from the other plant, a big B from one, and a little b from the other. Now, the next cross was the most significant cross probably in his study. He then crossed these hybrid plants to produce an F2 generation. And what he found in his studies was that red and tall, even though the original plant was red and tall, and its mate was white and short, red did not stick with tall in any predetermined pattern, and white did not stick with short in any predetermined pattern. He called this independent assortment. And what he got was the independent assortment ratio. 9/16 of the offspring were dominant for both traits. They were both red flowered and tall. 3/16 were red flowered but short. Another 3/16 were white flowered but tall. In other words, they were dominant for one trait but recessive for the other. That's these. So a total of 6/16 were either dominant for one trait and recessive for the second, or recessive for the first trait and dominant for the second. And lastly, 1/16 were recessive for both traits. Now, these two principles that I've demonstrated here, the first, that the fact that the letters for the plants separate, the fact that the plants get 2 letters for every trait, there's no blending, that was segregation, the law of segregation. Independent assortment describes the behavior of genes for separate traits as they're getting passed down. So if we did a cat study, and you'll notice this cat is pure for its traits, and this cat's pure for its traits. If you count cats up in the offspring, it's 9, 3, 3, 1 is the ratios you get. Here's the 3. There's 3 whites here. There's 3 of these tans here, and there's 1 cat that's very unusual, recessive for both traits. Now, Mendel took his paper and presented it at in a scientific society meeting in Brno, Austria in 1866. Unfortunately, the audience he was talking to believed in blending inheritance. Gregor was not famous, and the audience felt that his study was probably peculiar to pea plants, and that pea plants probably wouldn't be able to tell us anything significant about, animal heredity. So, but Mendel's basic principles were that the parents had chemical packets and they handed them to their children that remained separate and they did not blend. So every organism had 2 genes per trait, meaning we're diploid, we're not haploid. And every sperm and egg produced only gets 1 gene per trait. But he didn't understand meiosis, but that's because of meiosis. Mendel developed 3 laws in his paper. The law of dominance was that some factors are dominant and prevent the expression of other recessive traits. The law of segregation, which was the 2 factors for each trait separate while you're making your sperm or your eggs, so that sperm or eggs only get 1 factor per trait. And lastly, that the factors for different traits separate separately or segregate independently when sperm and egg get made. Those were Mendel's 3 basic laws. Now, there are a few exceptions to Mendel's laws, but basically, Mendel published at the Proceedings of the Brno Society of Natural Science in 1866. And because he was so far ahead of his time and because he was not from the aristocratic class or group, he had no influential backing and his work went unappreciated. So his paper just basically sat. Not many people read it. In 1900, Hugo de Vries, Karl Correns, and Erich Schermark rediscovered the same principles that Mendel had, and Mendel's paper was pulled, and he was given credit for being the father of genetics. The only problem was he had already passed away in the late 1880s, so he had unfortunately not enjoyed the fame and notoriety that he should have for being such a genius of his time. Today to do simple genetics problems, we use a simple procedure called a Punnett square, which is based on Mendel's rules for predicting the offspring of a cross. To do the Punnett square, you need to determine the parents' genotypes, and then determine the number of different, sperms and eggs that they can make, and then make a Punnett square that's going to be sized according to the number of sperms and eggs each parent can make. You write one of the gamete sets across the top and the other one down the side. And then you fill in the Punnett square, which are just combinations of sperms and eggs. And then you can write down the genotypic ratio in the offspring and the phenotypic ratio in the offspring from the Punnett square. The basic rules are listed here. We'll go over these when I go over the problems. But basically, you have to write down the genotypes of the parents, and the parents get 2 genes for each trait. You have to determine the types of gametes that each parent can make so that you know the size of the Punnett square that you're going to have to produce. Then you're going to draw a Punnett square that's determined exactly by the size of the number of gametes each parent can make. And you never repeat row or column headings within a row or a column. I'll explain more about that when we're doing the examples. And you write the sperm next to the row or column headers they represent and do the same for the eggs. And then you fill in the boxes of the Punnett square, which are sperm and egg combinations. You want to keep like letters together and put capital letters before recessive letters. So you want to write, these 2 are incorrect. TA is incorrect. Big T, big A, little a is just fine. But big T, big A is not fine. And little a, big A is not fine. Those are not following the rules of genetics. And then you really count up the number of each genotype in the grid, and you write down the genotypic ratios and the phenotypic ratios in the offspring. So there are a few exceptions to Mendel's rules, and they are incomplete dominance and codominance, which are like in human blood type. Turns out that blood type A is codominant to blood type B, so people who inherit the A and the B gene have the rare blood type of AB. So it is not always true that one gene dominates another. And sometimes when you cross plants with red and white flowers, you get pink offspring because the red is incompletely dominant to the white and you get pink as a result. There's another exception, sex linkage, and we'll talk more about this later. But it turns out that males, human males, especially mammalian males, are slightly genetically incomplete because the Y chromosome, which is homologous to the X, does not carry a full set of genes that it should. And as a result, males only inherit one gene per trait for all genes carried by the X chromosome. We'll talk more about that later.